I was reading an article about, I told you that sequencing projects are becoming, of course, I've read that this turkey just got sequenced. The turkey, the bird we eat on Thanksgiving. Well, you'll eat on Thanksgiving now that you're here in the United States. That is a genome that just got sequenced, and it's sort of a sign that as you're basically all commercially important organisms, are going to get sequenced. All scientifically important organisms get sequenced, and it's really just a question of the cost dropping to the point where people find it worth doing. When it costs $100 million to do the first, mammalian or higher order organism, you don't do that easily. But when it costs $500,000 to sequence the turkey producers get interested in that, and that's what we're basically continuing to see. Any questions about anything? Yes.
What is the importance of the turkey besides it was cheaper?
What is the importance of the turkey genome sequencing? As opposed to, so one thing you're interested, why would the turkey producers of the world care about sequencing the turkey genome? That I think is really what you're asking.
Okay.
So one thing that you want to study is, for example, what do you want turkey to do if you are a turkey producer? You want it to grow faster is one thing you would like it to do. You'd love to be able to turn 1 pound of corn into 1 pound of turkey. And so the question now of what genes are there, you look at, in other species, there are genes that are known to, control the rate at which organisms grow. Maybe you would like to look and say, how do I, what are the mechanisms that worked in related species? What is it that maybe I might be able to do? Maybe by changing a promoter sequence on this gene, I might be able to increase the rate at which turkeys grow and cut the amount of money it takes to grow them. So that's why the turkey, or maybe, turkeys are resistant, maybe a lot of them have problems with certain diseases. And you might now want to know, well, how do I, design a turkey? How do I breed a turkey? How do I find drugs that are more effective to sort of protect turkeys against diseases in the capacity, in the conditions that they're, grown for commercial purposes? That might be one interesting.
I guess the chicken was already sequenced?
The chicken was previously sequenced. And one of the things that they're clearly going to want to do is job one is to take what other birds had been sequenced. I believe the chicken's been sequenced. There are other birds, I believe, a few other birds that had been sequenced. Okay? And of course, one of the questions is, how do they differ? When did the turkey evolve? At some point, is a turkey, where does the turkey differ from a chicken? Okay? How much of that change has been from, in terms of, because of result of domestic breeding versus wild, the wild turkey? These kind of things you might be able to study in a meaningful way, given the genome. Any questions? Yes.
So when they say the sequence is complete, then does that mean that implies some errors?
Okay, so when they say that the sequence is complete, what does that mean? I interpret that as meaning the same thing it means when you say your homework assignment is complete. Okay? It doesn't mean it doesn't have errors. Doesn't even mean that you have answered all the questions. It means that you've turned in your paper. So it's a little tricky to know what you mean by a sequence being complete, okay? Because you come up with a problem, probably a lot like on your homework, where it becomes asymptotically, requires more and more work to make your paper a little bit more complete. And this is certainly the case with genome sequencing, that there are parts of it that we talked about coverage, okay? And how you need a high coverage to eliminate all gaps, right? Each time you have a higher coverage, you pay more for the higher coverage. And there is a trade-off between how much knowledge will you get from filling in these remaining gaps, okay? Fixing these remaining uncertainties. And it becomes a trade-off. You'd like it to be as complete as possible, but you also have a budget. And so when I interpret genome sequencing as being complete, it pretty much is when groups have stopped playing with it, okay? But like, for example, there are two groups that recently sequenced the cocoa plant. I think I mentioned that, right? And I was reading in Science magazine this week that there's a nasty battle between these guys because one side made a big press conference, said we did it, The other side said, wait, your coverage isn't very good. We have a lot better one, and we're not getting any credit for it because you guys have better press people than we do. So bottom line is, it's to a certain extent a question if it's in the beholder. Actually, where this is really interesting is I am on a committee. I don't know, have any of you ever heard of the X Prize? Okay, what is the X Prize?
I've heard of it, but I don't remember what it is.
Okay. The X Prize is this organization that runs these contest prizes for big technological things. So they figure that people work best when they have competition. And like there was an X Prize. What was there an X Prize in?
Into space?
Into space, right? How do we get companies to decide to go into space? We say we'll give you $10 million if you're the first company that can send us somebody into space. And once you have a contest, you need rules, right? And so you now got to define what does it mean to go into space. And for the X Prize, it means, well, you got to get 50 miles up or 60 miles up, and you've got to do it twice, so you know that you can really do it. Okay? There is an X Prize for the first group that can sequence 100 people in 10 days. Okay? And the question of how do you judge this thing? I was part of a committee that was put together by the X Prize Foundation to decide how do you judge when somebody wins the prize. And this is the big question is, what do you define as sequenced? It's the exact question you're asking. When are you done? And you get into worlds where certain parts of the genome are very, very, very hard to sequence because it's very, very repetitive. It's also very, very uninteresting. Okay? So you have to really define what does it mean to define the genome, sequence the genome accurately, completely. Okay? And the debates in the committee were basically about exactly this kind of a question. And the kinds of standards that they seem to have is they'll say, well, you've got to sequence 98% of the genome to an accuracy of at least correct all but one every 100,000 base pairs. That's and distinguish between the two haplotypes, the chromosome you get from mom and the chromosome you get from dad. Okay? And the trick is to try to define complete in a way that it's challenging, but complete in a way you can check that they actually do it, right? If you were say, okay, you tell me that nobody else in the world has ever sequenced before. If you come by and say, oh, here's a genome, I don't have a right answer to check it against, right? So it's a tricky question. How exactly do you judge these things? And but the real question as to whether the winner will be next week or 100 years from now is when how accurately is accurately enough. Okay? Any questions? Okay? Okay, that's the news. That's the recreational part. Any other questions about anything technically? Okay, so what I'd like to do now is just quickly, before I forget again, I'd like to go through and discuss the semester projects. Okay, so a couple classes ago, I passed out semester project lists. These are on the web. Here's the remaining ones. So circulate these and get them back to me at the end if you haven't. If you've already taken one, you don't need another one. But I want to talk to you briefly about what your project is. So right around Halloween, okay, I am going to make you turn in a proposal. This is a 3 to 5 page paper that makes clear you know what you're going to do your project on. It means you have done some reading. It means you have done some preliminary investigation. It means you have picked a topic. Okay? So I want you guys to start thinking about that. You'll also have to turn in a progress report and then the final report. Each of these will be graded, okay, to encourage people to not just do everything the 48 hours before the final report is due. Okay? Any questions? This is a bad idea for many reasons. One is, of course, you can't do a good project 48 hours before it's due. The other is you probably can't discover that whether you have a good idea of what you're doing or not, unless you start far in advance. So what I do for these projects, I give you ideas of where you might start that are about a paragraph long. you're going to have to flesh these out into real proposals and projects. And sometimes you will find that there is a project on this list, and it sounds great up until the moment you start working on it, right? You will discover, oh, he says do this. You use this data, but there's no data like this to be found anywhere in the world, okay? And then you have to change what you're doing, and you couldn't do that, okay, if you start 48 hours in advance. Does that make sense? You've got to figure out, is the question clear? Are the data sources really there? What is it to be done? This is what you're supposed to be investigating in your proposal phase, okay? So what I think I would like the goal of your proposal should be, you've written enough down, you've investigated enough about what you're going to do and plan it out, that you're pretty convinced, if worse came to worse in the last 48 hours, you would be able to do your proposal without finding an unexpected surprise, okay? Again, I want you to work more than 48 hours on it, so that's not what I'm trying to say. But you really have to make sure that you've got the data you're going to use and that you have an idea of what direction you're going to move in. Because with many of these projects, people will boldly pick a number from 1 to 14, which is the name of the project. They will turn in a proposal that is copied what I had here, plus the result of Googling the term, okay? And they'll feel okay about it. I won't give them too bad a grade. But they won't actually really know what they're doing, okay? I'll try to catch it. But you guys have to know that you know what you're going to do from then on. Any questions about that? Now recognize things might change, but it's important to do enough due diligence on the project to make sure you have a clear direction of where you will go. Any questions? Okay, so let me, what I will do is now, the projects, you guys can pick your own project topic if you find something is interesting. Let's say some of you may have an idea, I want to do a project on X, and X is not listed here. You can convince me of this, okay, if it's a good project topic. So I'm happy to let you convince me to do something else, okay? I'd rather it not be something that you're obviously reusing for another class, okay? Or not obviously be what your research advisor is telling you to do anyway, okay? But if you have a topic in mind, I'd like you to feel free to talk to me about it. The other ground rule that I would like to say, I like people working in groups, okay, of 2 to 3. That's my preferred way of doing this. And I'm going to insist that all the non-CS students, the life science students, which is what I define as non-CS, have to have in their team at least one CS person, okay? Last time I found that, first I think it's good to have the interactions, okay? Because I think that the CS people have different skills than the non-CS students. And I didn't really like the project so much that the non-CS people did by themselves. There was a certain, there was a cultural mismatch there. So I want to see, if you're a non-CS student, then I want you to make sure you find at least one CS person in your group. Any questions about that? Okay, good. Okay, let me tell you about what the topics are that I have in mind, I put down on the sheet. What I encourage you to do is to read them, think about them, maybe Google a little bit, and then come by and talk to me, or one of the other people who are listed as the associated person for that project to get into it a little bit more. Okay? Any questions about that? So let's go through these pretty quickly now. The first set of projects are, we had one of the outside speakers for our course was Michael Schatz at Cold Spring Harbor Lab. He desires to interact with computer science students. Okay, he is a computer science PhD. He's at this biology lab. And he would be very happy to have people come and work with him on some projects. He lists 3 different projects here, all of which are related pretty strongly to the kind of things that we have talked about in here. The first one seems about mapping sequence reads back to a genome. The second one is about, again, looking at, given that you've got sequence read data and a reference genome, look for find reads that match except for small insertions and deletions. Okay? Another one of parallel genome indexing. We talk about suffix trees, suffix arrays. Suffix arrays are basically an index of a large string, and that's the kind of thing I think he wants you to talk about here. He even mumbles the word suffix array or Burroughs-Wheeler transform, which is an amazing, closely related idea. It's both amazing, and it's amazing that it's closely related. So if you ever want something to Google Burroughs-Wheeler transform, because it's interesting. But so if you're interested in these, do a little bit of Google and thinking, and then mail this gentleman a polite letter saying, gee, we'd like to get together and work with you on this, okay? Preferably arrange your teams or something like that beforehand so he doesn't get an infinite number of individual emails. And you have to then convince him that he wants to let you work with him, okay? Any questions about that? So with the proposal stage, the wrong thing to say is, yes, I want to work with this guy, unless he has already responded that he's going to work with you too, and you're convinced that something's going to happen here. He may see you and decide he doesn't want to talk to you. He may find that he's no longer got the time to work on these. You've got to make sure that there's a match there. Okay? Any questions about that? Okay. The second project I had in mind was a project that I like, although it is probably something that a lot of people that we have this news analysis project where we've analyzed we have a lot of data of the type, how much is this disease being talked about in the popular press or the scientific press? And are they saying better or worse things with it over time? Okay? These drugs, is it being talked about in the scientific press and the popular press? And are they saying better or worse things over time? My belief is that that there is something to be said about how the world understands and interprets diseases, okay, by looking at these trends. And so what the good news is that we basically have the equivalent of, I think it's pretty much down to an Excel spreadsheet or something close, with a lot of news data about these things. And the question is now, understand enough, learn enough about different diseases and drugs so you can say something interesting about the trends and how the popular press sees things versus the scientific press, and so on. So the trick here is to learn a lot about what different diseases are, how they are associated with it. Do people talk about diseases that are highly lethal or highly prevalent, or just very sexy, okay, for some reason? This is the question you'd like to learn. And this is one that ideally someone who knows something about drugs and diseases would be the best person to start on something like that. Any questions about that? Okay, patent analysis. Biotech companies and the pharmaceutical companies value patents to a degree that is quite different than in the technical fields. Yes, every tech company gets patents, but biotech companies live and die by patents in ways that the computer science companies do not. And it would be interesting to be able to study things like how important is a patent, okay? That may be a function of is the topic it's about important. Do a lot of other things cite it? Does it have a lot of time till it expires? Stuff like this. And so I think that there's interesting projects to be done in taking the text of 3 million patents and trying to do some kind of analysis of them. So in principle, we have data on 3 million patents. Okay, what we'd like you to do is to go take them and do something interesting with them. Okay, and you have to figure out what doing something interesting is. And I would encourage you to start by doing some reading about what patents are and what kind of text analysis stuff has been done on patents. Okay, because there is a literature on this kind of thing. Any questions? That might be an interesting project. Charles Ward is my postdoc, and he has a couple of projects on stuff related to what we are interested in. And if you're interested, you should contact him. Last year, he had one project. That was the one that was, I think, the best one that any team did last year. Okay, ended up being a paper published on it. What were the projects? One is we do some work on designing genes under constraints. And there is a need for, there's an algorithmic issues of designing these genes under constraints. But there's also an interface issue. You would like to have a tool that will design genes under, to satisfy certain conditions. We need a proper user interface for this. And that's what the first project is. It's about building a user interface on top of gene design algorithms to do interesting things. And I think that this is potentially a good problem. The other is a data analysis problem. We've talked about the triplet code of DNA. The triples are called codons scientifically. We'll talk more about that in the next unit. But the statistics about how often certain pairs of these things occur in genomes that have interesting properties to them. And problem B has to do with doing an analysis of this, comparing all the genomes that have been sequenced, and try to determine what you can determine about the statistical distribution of these pairs. Okay, and that's one that Charles will go ahead. Any questions? The fifth problem has to do with PCR. Remember, we talked about PCR as a way to amplify sequences. PCR is a very, very important thing to do. Designing the primers in PCR is a very important thing to do. And there's this paper that I have seen where they had a primer design algorithm that both looked like it was important and looked like it should be easily improved by real computer scientists. So I would kind of say, I kind of put that aside as something that might be an interesting thing to do. Find the paper from this guy, look at what they're doing, see why they're doing it, and see if there's a much better way of doing it. My sense when I read the paper was that there was such things. Yes?
How do you define a primer?
A primer is, so what is a primer? Remember when I talked briefly about PCR. Actually, I guess let's, and we talked briefly about PCR. We talked about the idea that the way PCR amplifies things is you specify a short sequence here and a short sequence here, right? And it's somehow hybridized to the appropriate DNA strand, and then caused it to amplify between here back and forth. I didn't give you the details of that. Okay, I think if you read in the book, they'll give you a more detailed description of how PCR works. But the triggering specification are two short sequences you give that basically say copy between any place where these things bind. Okay, that's basically what PCR does. Amplify between where these things bind. If you know exactly what sequence you're trying to bind to, okay, then designing the reverse complement is not hard. That's trivial, right? But what if, let's say, you know that there are a large family of species, and you'd like it to amplify this gene in a lot of different species. The flanking regions are not 100% preserved between species, right? If you compare the turkey and the chicken, okay, they both will have a hemoglobin gene, right? And you might want to amplify the hemoglobin gene without knowing what bird you're dealing with, right? And so you'd like to try to find a primer with the property that whatever species you have, it will bind a little upstream and downstream of the target that you want, with a good enough reliability that you can amplify it, but with enough slack that it can bind to slightly different sequences in different species. And that is really what this is about, okay? Any questions here? You have to do a little bit of reading, but I think this is an interesting thing. PCR primer design is a good problem, and and there's a lot known about it. But degenerate primer design is an interesting problem. Any questions? So this has some algorithmic issues there. Any questions? This notion of hybridizing, where we had that certain sequences attached to other sequences, is used in this thing called DNA origami. There's a desire to sometimes make physical objects out of DNA. And in fact, there's a very, very clever way of making patterns of DNA, where they'll give you as input a specific viral DNA sequence. you design strands that basically make it hybridize, okay, in ways that sort of staples, if you wish. Actually, let's just think. Let's say you want the DNA to fold like this. What if I design a strand that to encourage this to hybridize to that? Okay. Actually, let me think if I can think about how I'm doing this again. Right. Suppose I want this DNA to fold like this, physically. Maybe I could construct a sequence of staples such that this piece will hybridize with that, this will hybridize with that, and because these structures, the way I design these staples, this hybridization will basically form a certain structure in the big outer sequence. I don't know if what I said make any sense. I have a big outer sequence, and I'd like to fold it in a way that it produces some kind of a structure. So in fact, when you read the papers on it, one structure that people might want to make is a smiley face. Okay, what is a smiley face? I guess a smiley face has, I'm going to think about it. Here's a smiley face, right? You could imagine building a structure that traces out the picture of a smiley face. Okay? And if you could get DNA to fold in such a way, let's not get rid of the eyes explicitly, right? Maybe you could add other pieces of DNA to encourage it to fold in this way. Does that make sense? You're given one big sequence. Your job is to design these so-called staples so that the thing folds to the pattern that you want it to. And you have these really amazing papers where you can look at this stuff under a microscope and you see smiley faces. Okay? The DNA forming particular shapes. Yeah?
What is the practical use of these things?
So the practical use of this thing is, first of all, it is amazing that you can fabricate microscopic structures to what you want. So the first thing is it's amazing. The second thing is it's actually potentially very valuable. Now, you guys have probably heard of VLSI circuits, right? What are VLSI circuits? They are microscopic patterns of things, okay? Structures that people have etched, in glass, right? Now, this gives you a new way to make microscopic sized patterns. Maybe you can use this thing as a framework to build circuits around or something like that. So this is one of these nanotechnologies that it's not clear exactly what the right thing is. But it's amazing that you can control matter on this scale, okay? And somebody will find the right thing to do with it, okay? So the question is really, how do you design a smiley face out of DNA? And are there better tools that you can make to do something with this, okay? Is there something you can do with this kind of self-assembly, okay? People use the buzzword is nanotechnology. Make things very small that have interesting shapes. This is a very interesting area. And you should look at Rothman's paper and see what he's doing, because it is amazing. Any questions? Number 7 might be a good one for people who care about protein folding. I know a lot of our AMS people care about this thing. There is a question of now, suppose I want a protein of a particular shape. Could I design a protein to fold into a particular shape? Okay, if you think about what this was doing, this was sort of a tricky way to get DNA to fold into a particular shape. We'll talk about protein folding later in the semester. The issues are quite different. But the question is, can you look at come up with a model or a technique to design proteins that would fold into a particular, you have a shape you want. There's certain forces acting on proteins. Can you design a protein to fold into the shape that you want? And there's some very nice computer science-y models that govern how proteins should fold. And under these, there are computer science-y algorithms, okay, for designing things that fold into that way. And I think this is actually a good problem for people, especially the AMS people who care about protein folding. If you find some computer science-y person, these are interesting things. So there's papers there. Start to look at them, see what you can do. Any questions? Number 8 has to do with sequence assembly, but with a sort of there are always new technologies people are trying to build for sequencing. And these things lead to different sort of algorithmic issues or problems. So one technology there's a company called Pacific Biosystems that is coming out with in principle, a new technology that's going to read individual molecules and take over the world. Their problem is that when an abstraction of their problem is that when they give you a sequence, a molecule to sequence, they will accurately read bases, but they will skip them randomly. Let's say I give you on half the time I give you the right base, okay? So maybe if my molecule was ACTGTTT, maybe a read might be ATCTT. Another read might be CGTT. Another one might be, ATCGTT. Okay? And the question now is, if I give you a bunch of subsequences of a target sequence, can you put it together to tell me what the sequence is? Okay? Any questions about that? So the input problem is basically I give you a bunch of subsequences with some other error. Okay? Can you reconstruct how many sequences do you need? What sampling density do you need in order to put it back accurately? Okay? Ideally, you would read about the PacBio machines and other technologies and try to come up with a good model of this. But basically, my question is, how well can you do with this? Many, many years ago, we looked at this. So a couple of years ago, we looked at this, and I think we built something that would do a pretty good job for very short sequences. But I'm kind of interested in something that you can scale and something that could do well in this. How well can you potentially do with that? Probably starting from where we were would be a good starting point. Okay? So my goal is to give you what we have and say do something. Okay? Any questions? The next one has to do with designing not proteins, designing basically DNA, RNA sequences whose RNA structure folds into the right place. It's sort of like the origami thing, except without the staples. Here as input, you were given a substrate, and you have to design staples. The question here is, I give you as input a protein. I want you to design a gene that would code for it, while the DNA of this thing folds into a certain structure. And that's what number 9 is about. Any questions? Number 10 is one that if you know any chemistry or just have building models might be kind of cool. You know, that one thing that people do in a lab is they use a centrifuge. Hopefully we'll have a lab tour at one point. You'll see centrifuges. What is a centrifuge? Does anyone know what a centrifuge is? Yeah, something spins. Why does something spin? Okay? Brings down things by weight, right? You have a mixture of stuff. You take a test tube of gook. In this gook is a certain amount of gold. Okay? How do you extract the gold from the gook? Okay? One thing you might do is you spin it around. Okay? And it will fall according to the weight of the stuff, right? So if you spin it fast enough, perhaps the heaviest stuff settles first, right? And the question is now, in principle, you could be able to extract layers from this thing. And people use these centrifuges to help separate compounds. And so my question really is, given the way centrifuges work, okay, can you come up with a strategy that if you have something you'd like to separate out of a mixture, whether you think the mixture contains this thing, what is the right centrifuge strategy you would use? How fast does it spin down? Stuff like this. And I think there is an interesting problem here. You have to understand how centrifuges work. You've got to understand what the problem is. You've got to tell me what the problem is. Okay? But I was asked this by a guy who knows something, who I'd probably put you in touch with. And I think there's interesting problems there. Any questions? The next one had to do with, there's a lot of ways you can get personally sequenced these days. Okay, so you can go and send a check off to some company and they'll give you some level of sequence data about yourself. And the question I'd like to see is, what could you do with the kind of data you have in these databases? Okay? The good news is there's lots of exciting things you can do with it. The bad news is I don't know if you can get access to any of this data. And the question is, can you figure out anything interesting, okay, about, paternity? Like, for example, you give a little of your sample, you get a little sequenced. Can they figure out where your ancestors are from? In the United States, this is a big deal, okay? There's all this data out there. I don't know if you can get it, but it's got to be interesting things you can do with it. So do interesting things with it. That's the project. Any questions? Under 12, I list several projects. Here what I'm saying is, we're going to talk in here about hidden Markov models. We're going to talk about a lot of things. I want you to build find other data mining technologies, like phylogenies. We're going to talk about that. What I would like to see you do is, if you can find a good application for a data mining model using either hidden Markov models or support vector machines or Bayesian networks or phylogenies, I think that would be a great project. Now, these data mining projects are good if and only if the following two conditions are true. One, you have an interesting thing you're looking for. Okay? So, gee, I'd like to be able to look at text and decide if the writing is good or bad. Okay? Yes, I'd like to look at people's DNA sequences and decide if they're good, honest people or liars. Those are interesting problems, right? Some of them may not be supported by the data, but what I want to see is something where you have an interesting application and a source of data where you can do something with it. If you come back to me and say you want to separate people from Martians, okay? You don't know if somebody's walking around, you want to be able to tell whether they're a space alien or a human. That's a great problem. If you can find data on aliens, okay, so that you can train something to do it, right? In the absence of data like that, it's not a good project, okay? So what you need to do is to find a problem and find data for it. Okay, if you can do that, problem 12 is a great project. If you can't do that, it's a bad project. Any questions? Problem 13 involves I have a couple of algorithmic problems on strings. Many of these have been given over the years to the algorithm reading group. And these have the property that they are open problems and algorithms that would be interesting to prove some theorem about. They are all on string algorithms, which I consider in the purview of this. The good news is they are well-defined problems. The bad news is that they're theoretical problems, and if you could solve them, you might be able to write a paper about it. The bad news is that unless you have an idea of what you're doing, you might stare at these things for the rest of the semester and not do anything. So it's important if you try one of these, you figure out how you can make progress, maybe experimentally, maybe do something like this. So these are good problems for someone who might care about algorithms if they can figure out what to do, okay? It's not enough to say solving P equals NP, great problem, okay? But if all you do is stare at it and you can't show any partial results, I don't have anything to grade you on, right? Okay? And so that's really what I want to see done there. Any questions? The last option that I put down is people can write survey papers. I don't really tend to like survey papers because, they're usually not very good or adventurous. And so historically, survey papers don't get the greatest grades in here. If you want to pick some topic where there is a computational problem important in biology, and you survey the technology and you survey what kind of algorithmic issues and software solutions are out there, and identify really what the problems are and how they might be solved, that's an acceptable thing to do. Okay? But I do want you to write your own words. I want you to discuss algorithms. I want you to synthesize something. And, in general, I like the other topics better. But if you can't find a good topic, If you really have something that you won't live unless you learn more about genetic linkage analysis, this could be a good thing. Any questions about that? So what I want you to do is to calmly think about these things, go up and start to play with them, talk to people, do Googling and stuff like that, and then come back with a proposal when the proposal is due around Halloween. Any questions about that? Okay. Any questions about the project? Okay. Yes.
You mark some problem with stars, and that means you like them better. So it will be graded equal.
Okay. So the question is, what about the stars? So again, I build this thing every year. The one phenomena that I always try to avoid, which no matter what I do, I find I can't avoid. Whenever I give these things on the list, there will be one project on here that I'll say, I don't know if I should give this one because it's probably not that good. And I will find that everyone in the class picks that one. Okay? So I have, in the past, used the star system to try to guide these things. The one thing I did this year was reduce the number of projects. Okay? So my number, if you look at previous years, I had a larger number on the list. And one reason is I tried to preclude these things. So there are not that many different choices. The stars are not as strong guides this year as they usually are. So it doesn't mean that if you pick, I don't say that it means that if you pick a non-star project, you will get a worse grade. Often there is some other danger. Like I say, for example, with the algorithm problems. There is a danger you won't find a good problem there. Okay? So that's why, for example, I don't star that one. But if you find a good problem there, it's an equal thing. Okay? So don't be scared off from any of these because I don't have a star on them. But make sure you have a direction that you're sure that you can do something. Okay? That's really the important thing, that when you turn in your proposal should be a plan of action to do something. And you should be convinced that it can be done to at least a certain level. Yes, I have data. Yes, there is a simple algorithm I can run to do something on the data, even if it does miserably. Yes, I have an idea for a more complicated thing, and maybe I can't quite finish it. Okay, but at least there's something going to be there. That's really what I want to learn from the proposal. Yes?
So for all these, especially the ones with where you need multiple people, do you have to find the group first and then approach?
I would encourage you to find your group first. I do not really want to be a matchmaker here. Okay? So I encourage you, it might be, if it's necessary, I will be, but I'm not really looking to be a matchmaker on this. I encourage you to find other people who you might want to work with on things. Maybe figure that out together. Or it might be, you ask me about it and maybe you then try to find somebody so I don't intend to be the matchmaker. But if you ultimately say, oh, I really love this thing, and if you find anybody else, let me know. Okay? Then I might consider that. Any questions? Any questions about the project stuff? Okay? So think about it, talk about it. We'll deal with this over the month. Any questions? Okay. So what I'd like to talk about now, unfortunately it took a little longer than I'd like, but it usually does. Okay? What I'd like us to talk about now is get back into sequence comparison. Okay? So last class we talked about exact matching for sequences. Okay? We talked about the Rabin-Karp algorithm and the KMP algorithm for string matching. And any questions about that, quickly, just to bring us back to this. Now, exact matching in strings is, a good and important computer science problem. It is less important than the problem of approximate string matching for biology. So the next several classes, we're going to look at algorithms for approximate string matching, sequence comparison. Because, again, the biggest thing people do with GenBank is they say, is my gene, now that I've sequenced a turkey, here I have a turkey gene, what does this turkey gene do? And you want to find out what gene in chicken compares to it. What gene in frog, has anybody ever studied a gene like this gene I just discovered in turkey? Okay? Doing that means finding a sequence that is similar to what I'm giving you, but different. A lot of the bases have changed. There's been a lot of mutations. There's a long distance from a turkey to a chicken, right? And the question is, can you find a longer distance between a chicken and a fruit fly? And a longer distance between a chicken and yeast? And the question is, finding similar sequences in all the organisms helps you understand it. That's the reason why approximate string comparison is the big killer app when it comes to biological sequence analysis. And so we're going to spend the next 3 lectures, I think, talking about how we do approximate string comparison well. Any questions about what approximate string comparison is?
So you're saying they approximately match?
So what we're looking at is a world where before, the problem I might have given you is, when we looked at, let's say, what might we use a suffix tree for? I give you the Bible. I give you a pattern, fish. And I ask, gee, where in the Bible do they talk about fish, right? Now, I'm going to ask you to talk about, I might give you, a word that is not spelled quite right, okay? Let's say I give you the word skina. And I ask, where is skina in the Bible? Okay, skina does not appear in the Bible, right? But someplace there is in the text a place where it's relatively close, right? Maybe they're talking about skiing someplace, okay? Maybe more likely they're talking about skin, okay? If I look at a translation, skin cow. I don't know what they do, okay? But there might very well be a string that is SKIN space A. That's actually not too far from SKIENA, right? You inserted a space here, you deleted the E, right? And that was 2 changes. And so it might very well be that a string exists in the text with a small number of changes. Certainly the string will exist someplace with the fewest amount of changes. And that's kind of what we mean by approximate string matching. I give you a pattern that is wrong, not the right thing, but we don't know what the right thing is. Find where is there someplace in the text where it almost matches. Okay? In the biological sense, here I give you the genome sequence of the turkey, right? Here I give you a gene in a chicken, okay? And I now want to know where is this gene, the homologue of this gene. Where does it occur in the turkey? And you'll discover, yeah, it exists over here. And what's more, the changes between chicken and turkey. Here are where the changes are. And so I can now say, how similar is it? What's different? Why did it become different? Okay? Any questions about that? Yes?
If they're not exactly the same, how do you know that they're just because they're similar? I mean, how similar is similar?
How similar is similar? Well, we need a notion to measure what similar is, okay? Now, it should be clear that certain things are similar enough. I mean, it's a good question. Now, when I read when I read in the internet, okay, I see an article talking about a guy named Skiena. Okay? That is, my name has 1 N in it. Okay? Are they talking about me or not? That's somewhat similar. It's a spelling mistake, right? And maybe that's close enough, right? In fact, most of the references there are probably a misspelling of my name. On the other hand, I mean, that's just because I have a very rare name. I don't mean to make it's too conceited here. This is not the whole point. Google Skiena and see what you find. Okay? But on the other hand, so in that case, it's close enough, right? But now if I change this to, so the answer is, in general, actually, when we talk about genes, we need to have a notion. That's maybe a better example. I guess I'm getting myself in trouble here. You have a gene in chicken or yeast. You want to find what is the gene that 100 million years ago had the same ancestor. That's really what the answer of close enough is with respect to a gene, right? It meant that in the ancestor of both species, when they diverged 100 million years ago, did they have a gene that has been conserved in evolution enough that we can tell that they're the same thing, that they made the same protein, that the protein probably did the same thing. That's really what similar enough is. And so our goal is to try to find a sensitive enough string matching algorithm so that we can detect even fairly distant changes. Okay? So the goal is to try to, detect similarities that are interesting, okay, by some notion of what it means to be similar. Any questions? Okay, so let's go back here again. So what do we know about? We're going to be interested in computing, instead of exact string matching, we want to measure the distance between two strings, okay? And the usual notion in biology is we care about that is because of evolution. There's a common ancestor, two branches of the tree, there's lots of mutations. What we have today from the two species is no longer exactly the same, but it still has the same function, okay? And you'd like to now go back and look and say, these two are similar enough. They had a common ancestor, okay? And then you can subject them to more interesting analysis. Any questions? In the context of computer science, you'd also see approximate string matching fairly often. One is in spell checking. Again, I told you if somebody typed in a misspelling of my name, Google, type a misspelling of somebody's name or a misspelling of a word, they will often say, oh, did you mean to spell it correctly, right? So that means that somehow they were able to figure out that when I typed in cat, K-A-T, okay, they'll probably say, oh, did you mean to search for C-A-T, okay? They seem to have a notion that this string is very close to that string, right? So spell checking is an example of approximate string matching. Another thing that happens a lot is file difference testing. How many people have ever used the Unix utility diff? Okay, this is if you're a computer scientist, you use Unix. Diff is a good thing. I give you as input one file. I give you as input another file, right? Maybe I give you, a term paper. Let's say I give you a term paper one student turned in, and a student that a term paper another student turned in. If they were bad students, what would we expect to have happened? One copied from the other, right? In which case we would expect a hunk of text here to be very, very similar to a hunk of text there. Is that right? So finding similarities between hunks of documents for plagiarism detection is very similar to this problem. Okay? Finding the differences between documents is also, that's why you use diff, is more for finding differences. If you have a bunch of versions of a file, what exactly changed? What's the shortest description of what actually changed? That is what diff does. And all of this is sort of in the same family of problems. Any questions about that? So is it clear why we want to do approximate string matching? That much I want to be clear. Okay? Any questions? So the issue comes down to, when we talk about approximate string matching, what we are interested in knowing is we need a measure of when are strings similar, okay? How similar are strings? And a reasonable answer is one that minimizes the number of changes or the cost of changes to convert one string to the other. Why do we think that cat is similar to C-A-T? Okay? Well, one reason is because K and C, okay, there's only one substitution to get back there, right? We might think, though, that it's more than just that. What if I had the word mat? Okay? Is mat the same distance to cat, C-A-T, as K-A-T is? Yes, if we count character substitutions. No, if we count normal people's spellings, right? C and K have similar roles in English, right? And so it might be more likely someone's going to mistake a C and a K than an M and a C. Does everybody see that? So part of the issue is we might want to measure the distance between two strings in terms of either the number of changes or the cost of the number of changes, right? Maybe we say a change from a C to an M is more expensive than a cost from a C to a K. We'd like our string matching algorithm to permit us this flexibility to add these kinds of things, okay? Any questions? Does that make clear? So the notion of what similar or difference is somewhat application dependent. But I'll claim that there are algorithms we can do for string matching that let us hide that in a cost function, okay? And then you can use the right cost function for your application. Any questions? Okay, good. Let's come back. Boom. Let's try. No, somehow. Okay, hold on. Somewhere here I've got a mouse, don't I? Yes. Yes, it. Boom. No. Sorry about this. I'm sure I have a mouse, right? Boom. Okay. So what we're going to be talking about when we talk about string matching are, for the most part, there are three main natural operations that you can use to transform one string to another. Okay? There are two of them are, I'll claim, a question of perspective. Actually, no, excuse me. One of them is substitution. Substitution means changing a character. Okay, so if we want to consider substitution, we consider the case like, okay, let's come back here, sorry. In substitution, we can consider changing a character to another character. So a single character change is what we call a substitution. So shot differs from spot by one substitution. Namely, an H got converted to a P. Does everybody agree with that? We can also think about changing one string to another using what we would an insertion. You can change the word ago to agog by adding a G in the middle. Does everybody agree with that? We can change A, B, C to A, D, B, C by inserting a D in the middle. Each character insertion we can pay a cost for, and insertion is an operation to change one string to another. Any questions about that? A similar operation is deletion. We can talk about deleting a character from a string to turn one to another. Deleting the H from our gives our. Does everybody see that? Okay? And so we can talk about doing the three most natural edit operations on strings are insertion, deletion, and substitution. Any questions about that? Are there any other operations other than character, insertion, deletion, and substitution you might think about doing as a text operation? Yeah? Swap. So let's say when you're typing text rapidly, sometimes people will type, dog as ODG. They go, oh, got too excited typing it, right? And they switch these two. If we think about that in terms of changes, you could say, well, I can convert this dog to this by first deleting the D and then inserting the a D over here. That would be two changes, right? Whereas we might view a swap as one error, right? Does everybody see that? So maybe it's overstating the cost of an adjacent character swap by letting you express it by insertion and deletion. The algorithms we'll use, if you want, you can extend to single character swaps with no real interesting complexity here. But that would give you a way to charge less for that. What other kinds of operations might people do on strings? Yeah? Case change. So a case change might be someone writes, instead of writing Skina with the proper capital S, they may write it lowercase, right? And you're saying that might be, what is that? That looks to me like a substitution, but one that maybe you want to charge less than full price for. Is that right? If you want to compare two texts. The right thing is to say, yes, I'm changing it by doing a substitution. But changing a capital S to a lowercase s shouldn't cost as much as changing a C to a K or a C to an M, right? Any other examples of changes of strings, operations? Yeah.
In sequences, sometimes they have to shift the whole sequence forward.
Sometimes what you're saying is you move whole blocks of text around. Is that what you mean?
Yeah.
Right. Like let's think about that plagiarism example where you took your friend's term paper, right? And to try to, you said, well, I'm going to put some originality to it. I'm going to move this hunk here, right? Now, in terms of character changes, it looks like there's been a lot of operations, right? But in reality, all you did was one change, a physical movement from one to another. So one might try to think about maybe that should cost only one step, okay? And we might think about algorithms for trying to figure out the number of operations where you allow bigger hunks to move around, okay? That's algorithmically a little bit different than what we're talking about. But basic idea is clear. There are different operations we might have. The three ones that we're going to talk about in here are basically character insert, delete, and substitution. Any questions? Okay, good. Let's move this thing down here. Boom. Okay. Now, when we talk about what the cost of transforming one string is into another, there are two different ways to look at cost functions, or namely in terms of either distance functions or similarity scores. Okay? So when you look at, maybe let me see if I know if this will work. How many people have seen that when you talk about a food in the store and they advertise it 96% fat-free, right? That's saying that, is that any different when you say it's 96% fat-free to saying it's 4% fat? Okay? Is there any difference there? Conceptually, no. Okay? But in a similar measure, we can talk about the similarity of things based by either how far apart they are or by how similar they are, right? Suppose you have a twin brother, okay? And you could talk about your twin brother. You could either say that your twin brother is 100% like you, or you could say that there is zero difference. Does everybody get that idea? So there are two different ways we might talk about sequence similarity. The way that computer scientists usually talk about sequence similarity is in terms of distance between strings. Okay? So we might describe the distance in strings by being the minimum number or the minimum cost sequence of insertions, deletions, and substitutions to transform something to something else. Distances, describing similarity in terms of distances, has certain good properties to it. One property is that distance functions have certain mathematical properties that we sort of assume in the world, okay, that are useful enough we like to define them about distance functions. We say that a distance measure is a metric, okay, if it satisfies these four properties that logically we think about. When we think about distance, usually we think about Euclidean distance, the distance from me to you. If I have a distance function such that the distance between any pair of things is always greater than 0, that's certainly true about the distance, right? Euclidean distance. You can never be minus 3 units from me. Does everybody agree with that? So the Euclidean distance satisfies the property that it's always greater than 0. Can two things have a distance of 0, distance of 0 apart from each other? The answer is yes, but basically it's because they have to be in the exact same place, right? So basically we have a property that says that the distance between two things are 0 basically if and only if they're the same. The exact same, right? That's true about Euclidean distance. The distance between me and me is 0, right? Distant functions tend to be symmetric, right? The distance from me to you is the same as the distance from you to me, right? And that's something that we assume about distance measures, okay? And finally, there is a property called the triangle inequality that we naturally assume. That if I say, how far is it from me to you? Okay? The answer is, it is always no longer than it would be going through some other point z, and then so as the crow flies distance between x and y is always less than or equal to flying first to z and then flying from z to y. Does everybody see that? In Euclidean distances, this makes a great deal of sense, right? Under what conditions is it cost the same to go from x to y through z?
Z is on x to y.
If z happens to be on the line, it's the same. But if z is off the direct as the crow fly distance, it's greater, right? If your distance function satisfies that, we say that it satisfies the triangle inequality. Okay? And if your distance measure satisfies all 4 of these properties, then it is what we call a metric, and we have the intuition that it behaves like normal distances, right? The edit distance on strings is an example of a distance metric. So we'll talk about the edit distance, the minimum number of changes. Okay? That has the property that it satisfies all of these properties. The cost to change, let's think about that. Okay? To change one string to another always costs at least 0 changes. The cost is 0 if the strings are the same. The cost of transforming cat to dog is the same as dog to cat. Okay, this may not be completely obvious, because an insertion in one is a deletion in the other. That, I guess, now is completely obvious, right? And maybe a little less obvious, but true, is that if you want the edit distance between cat and dog, there is no string, there's no string cow, such that it is cheaper to transform cat to cow and then cow to dog than it is to transform cat straight to dog. Okay? Edit distance satisfies the triangle inequality. And so we know that what's good about edit distance is that it behaves like a normal distance measure. Any questions? That said, the biological world tends not to describe strings in terms of edit distance. They will typically describe things instead through similarity scores. Okay, a score which gets higher, okay, the more similar you are. Okay? You might say percentage identity is an example of a score that a higher score is better, right? You're 75% like my twin, okay? And we're going to see in here, when we talk about the Smith-Waterman algorithm, there is a good reason why they use similarity scores instead of edit distance, okay, for certain things, okay, for subsequence matching, substring matching, okay? But recognize that there is a difference. Recognize that computer scientists like thinking about distances. Other people like thinking about similarity scores. And believe me, that the same basic algorithms can work for both measures. So from now on, until I get to the Smith-Waterman algorithm, I'm going to talk about edit distance, okay? But recognize that the same kinds of things say, oh, I want similarity scores. You can compute in this pretty much the same way. Yes?
Aren't they pretty much the same thing, just in opposite directions?
You're saying that, okay, so they're not completely so how are they different? Let's think why a similarity score might be usefully different than just percent identity, okay? Let's imagine a world where, I have let's say I have a rock here. I'm just going to try and make it out. Here I have a rock this rock is 50% similar to that rock, right? Here I have a big rock. This big rock is 50% similar to this one, right? Which one has a more important similarity to it? If I want to try to find a big thing that is highly similar, right? This similarity, you know, if you take a look at the string A, is half similar to the string AB, right? It's not so impressive, right? What if I have the string S, SKIENA, and SS, KKIIEENNAA, right? Both of them are half similar to each other, right? Which one is more interesting that it's similar to each other? This one, right? You agree with that? So somehow a similarity score can capture both the percentage identity and the amount of identity. Does that make sense? So sometimes you want to roll these two things up together, okay? And here a similarity score, okay, properly measured, can measure both of these things, okay? Does that make sense? Does it make sense that in certain situations, this is the more interesting match than that, okay? And it's a question of not just the percentage of identity, but it becomes a question of how similar and over how long a thing. Any questions? So similarity scores are better at detecting this thing as being different than that.
Just because it includes a different, an additional attribute.
It's a question of both having a lot that's, it's saying that having a long piece where there is a high degree of similarity is more interesting than a short piece with the same degree of similarity. Okay? And this is somehow better off captured by similarity scores. It changes both the amount that they're the same in terms of fidelity and the length over which it applies. Okay?
If we take the long thing and we find that it's 30% different, it means the same thing as the 70% similar.
Yes, so 70% similar is the same as 30% different. That much I agree with you. Okay? But what I want to say is that you can imagine a score, if distance only measures fidelity, right? You sometimes want to talk about how big are the things, as well as how similar are they. And it turns out that capturing scores will capture that idea better. And we'll talk about that. You'll see that when we get to the Smith-Waterman algorithm. Just recognize these people, the biology people, are different. They often want similarity scores. But algorithmically, we shouldn't sweat it, because Skinner tells you that similar algorithms will optimize both things. Okay? Me. Okay, thank you. Any questions? Okay, good. Okay. So let's look at an algorithm now for computing edit distance. Okay? Or computing the edit distance. And I'd like us to look at this one algorithm which will give you which will tell you, given two strings, what the fewest number of changes there are between those two strings. We agree that the changes we have are of three types. Okay? Insertion, deletion, and substitution. There is an algorithm, a string algorithm, alignment algorithm, which will figure out the minimum number of the minimum cost sequence of changes using the observation that if we're converting this character, if we're converting this string into this string, we're going to have to do something about the characters on the right-hand side of the string, right? If we are converting cats to cats, something's going to happen at the other side of the string. Maybe we're going to decide that the S and the Z are related by a substitution. Maybe we're going to decide that we should delete the Z from this string. Okay? And then the cheapest way, in the minimum number of changes to convert it to this thing. Maybe we will delete from that. Maybe we will insert on top. The claim is that if you take, the big claim here is if I have one string S and one string T, and I find the minimum sequence of changes to do this to this string, something is going to be done with the last characters of the string. And it's either going to be that they match. It could mean that that they are, one of them is deleted. It could be that the other is deleted. Okay, unfortunately, I didn't. Boom, boom, boom. Right? These are the three possible things that happen at the end of the string. The cost of a match or a substitution. If they match exactly, I shouldn't pay anything, right? Because there's no change cost in that. If they differ, there's a substitution of one character. The cost of deleting this is one character. The cost of inserting there is one character. We could end our alignment with any one of these three things. The claim is that once we make that decision, the total cost of the job is the cost of that last change plus the cost of aligning the rest of the strings. Okay, so we're going to pay the cost of the last move plus the cost of solving this smaller job on the rest of the strings. And this, I claim, you can compute recursively. What is recursion? It is a problem of solving solving the same type of problem on a smaller input. If I can give you the answer to the big problem in terms of the smaller problems, then basically what is the recursive way of solving it? Solve the smaller problems. Okay? Get the answer, and then use that to make the decision for the bigger problem. That is the idea of using recursion. The key point to see here is that I know on the right side my optimal alignment will end in one of three ways, because it can only end in one of three ways. And the best way is going to be the one that minimizes the cost of doing the last change plus the cost of doing the rest of the strings. And if you see that, you get a recursive algorithm. Let's now zoom in on the recursive algorithm. Is there a way to zoom in? How do I zoom in here? Okay. I think I have a preview where I can't zoom in. Is that right? Anyone know how I zoom in here?
It says view on the top.
View. Zoom. Boom. Let's go view. Zoom. Boom. Let's look at this program. No, this is somewhat painful, but let's try that thing. Here I claim is a program written in C, so a genuine program, that will compute the minimum number of changes it takes to convert one string, the string S, okay, into a string T, okay? So I'm given as input two things. I'm given a string S, and I'm given a string, the two strings I want, string S and string T. And I am going to say that they, as far as I'm concerned, end in the ith character. The first string S ends at position I. The string T ends at position J. Does everybody see that? What I'm going to say is, I want to find what is the cost of transforming S into T. And I claim that there are three possible things I can do. Okay? I can end on a match. If so, what do? I have to pay the cost of matching the ith character of S to the jth character of T. If I do my alignment that way, I pay the cost of the last character match plus the cost of finishing the job on the rest of the string. And the rest of the string is S going from position 1 up to I minus 1, because I've killed the last character there, right? And on T up to J minus 1, because I killed the last character here. So this is a recursive call that computes the costs. It's recursive. It's the same function name, right? And it says the cost of ending on a match is the cost of that last character match plus the cost of recursively editing S to T for the